This is Dr. Matthews again. We're going to talk about bacteria and archaea. First, we're going to talk about shapes. They're identified, we identify these organisms by the shape they're in. Cocci are round. You have many cocci, one coccus. So that's terminology correct. Bacilli are, you have multiple ones. These are your rods. And a lot of people in a clinical setting will just say, oh, we've got some rods. They mean bacilli. They're rod-shaped. And you have bacilli is plural, bacillus is singular. Rod-shaped, some form endospores, which form a hard casing around that bacterium so that it is safe from the outside environment. Some are pleomorphic, which means they're basically not really distinct. They may be look kind of like a bacilli. They may be elongated, spherical. And this is a bunch of different kinds. This is cocci, one coccus, multiple cocci. This is a bacillus, a bacilli. Vibrio, see they're curvy. Spirelli, they're spiral shaped. This is pleomorphic. This is all one organism, but look at the different shapes. This is a spirochetes, it's curlicued. Now diplo just means two stuck together. So if this were a coccus, it would be diplococcus. If it were a bacillus, it would be diplobacillus. Now staph is, or staphylo. Staph is Greek for cluster of grapes, and you can see it clusters together like grapes. So if this is Staphylococcus and Streptococcus, these are strings. Think of a strip for strep. You can also have Streptobacilli, which is a whole long string of bacilli. We have some pictures of those somewhere. Bacteria can be in palisades, which are these just lined up together. They can be in tetrads. The most common things you will see in a lab will be cocci, bacilli, and then staph, strep, diplococci, and the pleomorphic organisms. To me, these are not the greatest pictures. They're the ones your book provided, but this is Staphylococcus. We have some better pictures. At least for the lab section, some I took myself. But you have clusters of staph. They're a little hard to see that they are cocci. This is Staphylococcus aureus. This is Escherichia coli, commonly known as E. coli, and I say it's because no one can spell Escherichia. But you see it's bacilli. These are gram-negative. They stain pink. These are gram-positive. Over here they stain purple or blue. And here's a pleomorphic organism, Coccobacilli. And these are gram-positive because they're purple looking. And see, this is different shape than that one, but they're all one organism. We have some Vibrio, very difficult to see, but they're curlicue. Spirillum, again, I don't really like that picture, but I kept this slide because of this picture, the spirochetes. You can really see this with a black background and the curlicues. And some of you may be familiar with the most common spirochete that we talk about in this class is it's going to be syphilis. Okay, diplococci. These appear in pairs, and a good example, Streptococcus pneumoniae, and Streptococci, Streptococcus pyogenes. They have little bead-like chains. Tetrads, it gives no example of these, but there's 4. Sarcinae are cubes of 8, and it's a result of the dividing and sticking together. in division in 3 different planes. Now, Staphylococci, grape-like clusters, and there'll be just glops of grape clusters here and here and here on your slide. So here we have staph, streptococcus, and then micrococcus, which appear to be tetrads. See, 1, 2, 3, 4. 1, 2, 3, 4. Diplobacilli are paired rods that remain in pairs after division. And streptobacilli, they stay in chains. Bacilli in chains. It sounds like a heavy metal band. And here we actually have some streptobacilli. And here we have some diplobacilli. Just 2 together. Bacteria multiply by binary fission, much more simpler than meiosis or mitosis. Just the DNA replicates itself, the bacterium elongates, so it goes through a period of growth and elongation. The DNA goes both directions, it splits down the middle, and now you have cytokinesis. This is splitting. You have 2 daughter cells. This is the exact same cell as the original was. Then these are going to elongate and do it again. And in a typical bacteria under typical good environment, it maybe takes 20 minutes for this to happen. So you have 2 now. So in 20 more minutes, you're going to have 4. 20 more minutes, 16. So they multiply very fast in a logarithmic fashion. Now, when you have bacterial growth, or when you have growth of any organism in an environment, we're going to first think about bacteria in a petri dish. First thing that happens is nothing. It gets adjusted to its environment. So that's the lag phase. Then they grow exponentially. Then if left alone, they'll use up all their nutrients and they sort of start slowing down, and then they go through a death phase. Now this is found to happen in many organisms. If you get too many rabbits and there's no predators, they'll do just this. They'll eat up all the grass, they'll die off. Wonder about humans, if we're not a little bit overpopulated, causing some of the issues we have going, the viruses, plagues, death, fires, and all that type of fancy stuff. Okay, measuring growth, you can measure the dry weight. This is the most accurate, time-consuming though. Turbidity or cloudiness can be numerized. You can put a number on it by putting a little drop on a spectrophotometer, and light passes through and it can calculate the mass per unit. You can get an idea of how much bacteria is in that drop. You can do direct cell counts, and let me show you these chambers. You have these little chambers that they put the bacteria in. You can grow them in these, or you can put them on a slide, certain dilution solution like you had in a liquid, drops, and you count them. And chambers like these may be why I got out of research, because I was working in parasite research. And when you get to number 523 on worm eggs and you lose count, it's very disconcerting. Things that are going to influence bacterial growth: nutritional requirements, temperature, osmotic pressure, other atmospheric conditions like heat, cold. Now, nutritional requirements. Where do they get their energy? Phototrophs. Troph refers to eating. Phototrophs get their energy from light. Chemotrophs get their energy from chemical compounds. Now, carbon is the backbone for all organic compounds. This used to would have read for all living organisms, There have been some arsenic-based organisms found in the bottom of the ocean, so we can't say all living organisms, but we are carbon-based, and everything that we see and know is carbon-based. We have autotrophs that get their carbon from the atmosphere, and heterotrophs that use their carbon from organic compounds. We are heterotrophic. We can't just absorb carbon dioxide from the sun and from the atmosphere and sit out in the sun and photosynthesize. We got to eat something. So hetero means you got to get it from organic materials. Troph means eat. Photoautotrophs. This is a lot of terminology here. This is one thing you may want to print off, or you can just mark them in your book. That's what I like to do. The photoautotrophs use sunlight as energy and carbon dioxide from the atmosphere as their carbon source. This is the most typical cycle of life. Photosynthetic bacteria, algae, and green plants form the basis for our existence. A cow eats the grass, we eat the cow, that kind of thing. Chemoautotrophs use chemical compounds as a source of energy and then carbon dioxide as their carbon source. So they're taking atmospheric, or if it's in the bottom of the ocean, they're absorbing carbon dioxide from the water and using chemical compounds for their energy source. Photoheterotrophs use the sun for energy, organic compounds for the carbon source. Chemoheterotrophs use organic compounds for both energy and carbon. This is stuff that you're probably going to see at the bottom of the ocean. You're not going to just get these every day. Nitrogen. Nitrogen is essential for all living organisms that we know about because it's important as part of your protein, and it tends to be a limiting bacteria. The atmosphere is absolutely full of nitrogen, but it's not in a usable form. Now, some bacteria can take inorganic compounds such as nitrates and get their nitrogen from that. They take atmospheric nitrogen and get their nitrogen from that, and that is good because they take and transfer atmospheric nitrogen into nitrogen that goes into plants, and that allows life to go on because we have to have nitrogen to make protein, and we can't just make proteins from taking a nice deep breath of air. Sulfur and phosphorus, the need for them can be met by taking in organic compounds that have these in them, or from salts that have them in them. And then minerals are required for every living thing in trace amounts. Okay, temperature, and there's a lot of terms on these. Mesophiles, think of medium. That's human body temperature, and most bacteria fall into this classification. Thermophiles, these like hot stuff, hot springs, and a lot of these hot spring so-called bacteria are now classified as archaea. Haystacks, this is interesting, or compost pile. If you put your compost pile too close to the house, it could get hot, and if it gets hot enough, it could catch on fire. Same thing with hay. Hay that's too wet has been known to burn down barns because it gets hot inside as it's rotting, and it gets hot enough to catch the dry part on the outside on fire. There goes the barn. Cryophiles grow at very cold temperatures, 0 Celsius, freezing or lower. Psychotrophs grow very slowly at 0. They have an optimal range of growth at warm temperatures. So this is very common, and this is why we put food in the refrigerator to keep things from spoiling as fast. But things can still spoil at freezing temperature. They can still spoil at sub-freezing temperature. You don't want to take something dated 1972 out of your freezer and eat it. Osmotic pressure. Cell walls allow organisms to hold up to a lot higher osmotic pressure than things without cell walls. Think about this. Take a slug. Don't do this really, but think about a slug. You pour salt on him. He melts because of the osmotic pressure exerted by the salt, which creates a high hypertonic solution. Now take a plant, a living piece of plant, pour salt on it, and watch it for a few minutes. Let's take the stem of the plant. Let's don't do the leaves. And it will probably sit there for a while. Now if you pour the salt on its roots, eventually it will kill the plant. But if you pour it just on the plant stem, it's not going to shrivel it up rapidly. So osmophiles, they like high solute concentration. Halophiles, salt lovers. Like in the salt lake, there are organisms growing. Hydrostatic pressure, air pressure. Some organisms are barotolerant. They can live at high pressure. Some are barophiles. They only grow at really, really high pressure. So this is at the very bottom of the ocean. If you took those organisms out of the bottom of the ocean, they would probably explode. They certainly would die. Okay, aerobes and anaerobes. Aerobes have to have the presence of oxygen. Obligate means they're obligated to have oxygen. They must have it. Obligate anaerobes cannot grow in an oxygen-rich environment. Gas gangrene is a good example. You have a crushed piece of tissue, maybe a foot or leg, they're not getting any oxygen to it. The organisms, which would be generally some sort of clostridial organism, will get into the tissues and they start to grow because it's a perfect condition. And part of the treatment for this, just giving them antibiotics isn't going to do anything because there's no circulation if you have no oxygen. But putting them in a hyperbaric chamber may help to kill the organism off. Okay, aerotolerant anaerobes. They can exist with oxygen, but they don't like it. They have to ferment. They don't burn oxygen. It just sits there. Facultative anaerobes, they're like a switch hitter in baseball. They can use it or they cannot use it, oxygen. Microaerophiles, they need just a little oxygen. Catnip bacteria require a lot of carbon dioxide, more than in a normal environment. Now the pH is important in growing organisms. Some things, soils can go in lakes anywhere from 0.5 to 10.5. There's different ranges that things can grow in, but neutrophiles, these are things that are close to neutral, 7 being neutral, 5 to 8. Acidophiles below 5.5, alkalophiles above 8.5. Now urinary tract infections just love alkaline urine, and it greatly increases the chance of urinary tract infections. And in fact, those bacterium actually will alkalinize the urine more, making it a better environment for itself. And then you can, if it continues to go, that alkaline environment is a real nice place for stones to grow, kidney stones. And when they grow, then they get bigger and bigger. And in animals, dogs and cats, they actually make food, and that's all you can feed them, just the food that acidifies the urine, and it will help decrease the incidence of kidney stones. It will help decrease the incidence of urinary tract infections. People take cranberry juice for that. I don't know if it really works. We're going to talk about the classification of microorganisms. Prokaryotes are divided into bacteria and archaea, and they had to be separated out because there's such a chemical difference And the bacterium are just the peptidoglycans that make up their cell wall, just have nothing to do with archaea. They're just chemically totally different. The DNA is totally different. Everything's different. A lot more is known about bacteria, and archaea, this is a little bit of a misnomer. They're a group of, it should say, bacteria type stuff that have other features than bacteria. Spirochetes, helical motile bacteria, they have twisting motions, and they're generally in aqueous environments or in human or animal hosts. Treponema pallidum, syphilis. Borrelia burgdorferi is Lyme disease. Leptospira interrogans, and there are several leptospiras. They cause leptospirosis, which can cause kidney failure. And you get this from drinking unclean water where animals have gone wee, animals that are infected. So you don't want to drink water with animal pee in it, rat pee. And hopefully you won't, but if you drink out of a creek, you might. There's a leptospirosis organism. Gram-negative aerobic rods and cocci. And these are the cocci that's a little hard to see. Neisseria, this is Pseudomonas. These are bacilli. And they're gram-negative, they're pink. Now, there are many anaerobic, facultative anaerobic gram-negative rods. It means they can take it or leave. If you put a little oxygen with them, it's not going to kill them, that live in the gut. The most common is E. coli, but there are many, many organisms. Now you hear E. coli, oh, they're dying of E. coli. Well, just because you have a genus and species does not mean you have the strain. If you watched my previous slides about the genus and species, you saw that we have different varieties. You know, something's called a cat. Well, this was still, there were different genus and species. But, you know, you have a cat and a lion. I think a better example would be a Chihuahua and a Great Dane. The Chihuahua might be the normal bacteria, and the Great Dane or the Pit Bull or just any dog you want to choose that might bite you or knock you down would be the bad E. coli, pathogenic, and these come from animal waste or human waste. This is E. coli, gram-negative rods. This is Proteus vulgaris. It's a rod, and this, if you can see it, you can clearly see that that's a bacilli, but some of these are turned up on their end and they look round. You have to look at the bulk of them. This is another one. See, this looks round. Well, if you take a long, tall jar and you look at it from the bottom, it looks round, and that's what has happened with this cell right here. Okay, anaerobic gram-negative rods, they can be many different types, but Leptotricha and Fusobacterium are some we're going to mention. They grow along the gum line and tooth surfaces. If you don't get your teeth cleaned, they get under the gum line, and since they're anaerobic, they just grow beautifully. If you get tartar buildup, there's places that oxygen can't get to, and they start to grow. And they get down into the gum line and they start to separate the gum from the tooth, and eventually your teeth can fall out. And furthermore, if you have rotting, disgusting, bad teeth, bacterium can go from those gums, which are going to get cut by that tartar, and when you eat food, it can get into your bloodstream and infect other things such as your heart. Anaerobic gram-negative cocci, again, gingivitis. This is a big thing. They're found a lot of places: mouth, intestines, vaginal tract. But gingivitis is a big thing. So take care of your teeth. Brush your teeth. I just ate chocolate and hadn't brushed my teeth yet. But Brush your teeth after eating and before bedtime, and anytime you want to brush your teeth, brush them and get your teeth cleaned on a regular basis. Rickettsias and Chlamydias. These are very small gram-negative bacteria. For a long time, they were not considered to even be bacteria because they're so small. Now, chlamydias do not require an invertebrate to grow. And you know about the chlamydia that causes the STD chlamydia, but there are some chlamydias that infect eyes. There are many different species of chlamydia. Now, Rickettsias, and there are a lot of species of these, have a vertebrate host and an arthropod host. So these are going to be a lot of your tick-borne diseases. Rickettsias. That's easy if you can remember the genus name, you can remember the species name. Rocky Mountain spotted fever. And then Chlamydia trachomatis is urogenital infections. And this you can barely see a rickettsial organism. Generally, when you test for rickettsial organisms, they're so small, a lot of these you're going to look for just with a test kit. Like if I'm looking for Lyme disease or Rocky Mountain spotted fever, I'm going to just order a tick panel on my patient, and they're going to check it with an antibody test. Okay, Mycoplasmas and Ureaplasmas. These are tiny, tiny free-living bacteria. Now, unlike other bacteria, they don't have cell walls. They are pleomorphic. They're resistant to all kinds of stuff. Penicillin, cephalosporins, vancomycin, because these drugs act by interfering with cell wall synthesis. And since they don't have cell walls, they don't work. They can cause some diseases in humans, animals, and plants. We don't have any examples right now. Gram-positive cocci. We have aerobic and anaerobic organisms. Many are harmless. Many are very virulent. That means they can make you dead. Micrococcus, Staphylococcus, and Streptococcus highlighted, Peptococcus. Now, Staphylococcus can be normal flora in your throat. It's just nothing wrong. It's just there, as can strep. But a tiny little mutation in it can be something that causes meningitis or MRSA or many other things that kill you. This is Staphylococcus epidermidis. Now, all you can tell from looking at this slide is that it's a Staphylococcus. See, this it's a cocci, lots of cocci, and they come in clumps. As far as getting it down to specifics, you have to do other things. You might have to do an acid-fast stain, many different stains. May even have to get down to the DNA testing. But this, I know it's this because it's labeled on this picture. Endospore-forming gram-positive rods and cocci examples: Bacillus, Clostridium. These are really important in the medicine and the food industry. The spores are resistant to heat and disinfectants. They're widespread. Examples of Clostridium: Clostridium botulinum causes botulism. Clostridium tetani causes tetanus. These are things you don't really want. And if you don't, certain foods such as like home canned green beans, if you don't heat them hot enough, they're gonna, the Clostridium, if they happen to be in there, and they probably will be because it's so common, are gonna grow and just, they're invisible, tasteless, and the Clostridium itself doesn't kill you. The toxin it produces kills you. So if you have botulism, if they can keep you alive on a ventilator until you get over it, you'll live. That's one good thing about it. It's a bacillus. I don't know why they just have a random bacillus here. Endospore-forming gram-positive rods and cocci continued. Clostridium Perfringens, widely distributed in the environment. Any kind of decaying matter in the guts of animals, plants. Now, common cause of food poisoning, and most books just stop there, and it just kind of flips me out because they also cause gas gangrene. If you get Clostridium perfringens, or many of the Clostridiums growing in a wound that has got inadequate circulation to it, you're going to get gas gangrene, and that's very disconcerting because then you start losing limbs in patients. But they're generally free-living, obligate anaerobes, so you're not going to see them on the surface out in the air. But Clostridium difficile, that's an interesting one. I didn't put the whole thing. It grows as normal flora in your guts, and it doesn't bother you because there's so little of it, because E. coli is non-pathogenic. E. coli is the main thing living in those guts. So you take a lot of antibiotics, kill off all the E. coli, all the gram-negative stuff. Clostridium difficile starts to grow and multiply, and now it's run wild like a weed in your guts, and it causes screaming diarrhea, and it's a really bad thing to get. I had a student who said she got it from a patient. She gave it to her husband, and her husband did not notice the symptoms until he was on the up escalator at the Macon Mall. And then it happened all at once. And that was bad. All they could do is get on the down escalator and go home. Regular non-sporulating gram-positive rods. They can be obligate or facultative anaerobes, and they have different nutritional requirements. And we're going to look at Lactobacillus and Listeria on our next slide. Lactobacilli are used in yogurt, cheese, sauerkraut, pickles, and I love yogurt and cheese. I don't like Greek yogurt, but they're generally, most species are harmless, in fact probably helpful in cultures that eat a lot of yogurt or a lot of any kind of processed, any of these three. Too many pickles are too salty, but people tend to have healthier guts. Now, whether those bacterium actually live through your digestive tract or just make a healthier environment for the normal flora, there's no way of saying. Now, Listeria monocytogenes is a causative agent of listeriosis, and this comes from animal feces, and it contaminates food, and so there's been several cases of it contaminating things like meat, like sliced meat you buy for sandwiches, hot dogs, and I can't imagine how you get animal turds into a cooked hot dog. But what has to have happened is someone has gotten their hands dirty and then handled that food before it's packaged, because they cook these hot dogs, they cook this meat, or possibly they had this growing on their production line. But if most of us get Listeria, we're going to be fine. But if you're pregnant, it can infect your baby and cause you to have a miscarriage. If you're very old or very weak, it can cause neurological problems and death. We've got a few irregular non-spore-forming gram-positive rods, just a few examples. Actinomyces. These are not generally going to bother you, and they're opportunistic. You're already down, you're sick, and you have an infection, and they just jump in there and join the party and make it worse. Corynebacterium diphtheriae. And it is making a comeback. People aren't getting vaccinated for it like they once did. Propionibacterium acnes. This is impossible if you have this species name on it to miss on a multiple choice test. And then this actinomyces is another thing that causes gum disease, but also lung infections. Mycobacteria, gram-positive, aerobic, acid-fast. Now this is a second stain, so you'd have to do a gram-positive gram test. It'd be pink. Then you do an acid-fast test, and they'd be purple on that. They're obligate intracellular parasites. You can't just culture them. You'd have to culture them in a tissue culture. And even though they tend to look gram-positive, they are technically not truly gram-positive or negative. Resistant to many bacteria. Mycobacteria tuberculosis comes to mind. Methanogens, these are fun to talk about. And these are technically archaea rather than bacteria. Anaerobic, they grow in swamps, marshes, hydrothermal vents, and in guts. These are what put methane out into the air, and there have been recurrent things, I think it was maybe Switzerland, that put a flatulence tax on cattle. And the farmers did not particularly like that, and it did not last long because they said, well, that's causing global warming. Well, they should put a flatulence tax on everyone because everyone produces methane. And methane is flammable. Methanogens are killed by oxygen. Now, if you have teenage boys around and they have a lot of flatulence, you may find that they experiment to see whether they're flammable. And that's, I don't know if we have, maybe teenage girls do it this day, but it was always a problem in my household. I had houses, used to have a house full of teenage boys. Just everybody dumped their kids off on me. And I had to hide my lighters. Okay, that's the end of this one.